Hello, everyone. Welcome to the webinar. I'm Ann Thayer, contributing editor to the CNN Media Group, and I'll be moderating today's event. This webinar is entitled Recent Advances in Library Synthesis to Accelerate Medicinal Chemistry Research and is being sponsored by WuXi AppTec. CNN works with sponsors to identify topics of interest and value to CNEN's audience and consistent with CNEN's mission to provide news and analysis of the chemistry enterprise in a timely, accurate, and balanced fashion. Just a few technical items before we begin. During the webinar, you can adjust the size of the slides on your screen by grabbing the lower right corner with your mouse. If you need technical assistance, please look at the Help widget at the bottom of the screen or type your query into the Q&A box. If you're disconnected during the webcast, please log in again according to the instructions you received earlier. You're also encouraged to contribute to the success of this webinar by asking questions at any time during the presentation through the Q&A box on your screen. Questions will be handled at the end of the presentation, as many as time permits. If we don't get to your question, the speaker will have the questions in hand and will be able to follow up with you later. Please note that CNN does not endorse any company, products, or services that may be mentioned in the webinars, and that each webinar will be archived at CNN Online after the live webcast. Also joining us today is John Lord, Director with WuXi Discovery Services, and I'm now going to hand things over to John.
Hello, everyone. Welcome to today's webinar. I'm John Lord, Director with WuXi AppTec Discovery Services, WuXi AppTec, and I will be your moderator for today's presentation. This webinar is entitled Recent Advances in Library Synthesis to Accelerate Medicinal Chemistry Research. The presentation today is being sponsored by WuXi AppTec, a global company with operations across Asia, Europe, and North America. WuXi AppTec provides a broad portfolio of R&D and manufacturing services that enable the pharmaceutical and healthcare industry around the world to advance discoveries and deliver groundbreaking treatments to patients. WuXi AppTec's open access platform is enabling more than 6,000 customers from over 30 countries to improve the health of those in need. Today, we are pleased to have Yan Zhu, Executive Director, Head of Library Business Research Chemistry Services at WuXi AppTec. She will review with us the concept and application of parallel medicinal chemistry library synthesis, how PMC can provide more compounds with limited scaffolds as a more focused approach to target molecules, and lastly, help to understand new protocols to guide the follow-up of singleton compound synthesis to improve efficiency. And with that, I'll turn it over to Yan.
Hi everyone, I'm Yan Xu, Executive Director of Chemistry and worked in WuXi AppTec since 2001 for library synthesis. So I almost witnessed the change of library in past 23 years. Thanks a lot for your attendance for this webinar. It's my pleasure to share with you about recent advances for library platform and how library technology can accelerate medichem research and enhance chemical space exploration. In the first part, I will introduce the importance of library technology in drug discovery. The changes and recent advances in library platform include virtual library, library enrichment, parallel medicinal chemistry, kit screening, HTE to provide vast reaction data, and applying library technology in spatial area. For example, Covalent library and PMC library to accelerate PROTAC research. Library is well known to serve two main purposes. One is to accelerate hit finding, lead finding, and lead optimization. Library can quickly provide vast collection of diverse compounds for testing, save time and resources. It is an essential part of drug discovery process for big pharmas and more widely used by biotech as well. Secondly, is to enrich compound collection. Library synthesis is the process of synthesizing a series of compounds and evaluating them for hits that have desirable therapeutic properties for a given target. It can allow pharma companies to do efficient and systematic screening of biological pathway or target classes. Improving the success rate of drug discovery, the compound collection of existing library can also be screened against the novel targets to find the hits that have properties that could lead to new therapeutic uses. Nowadays, library technology is more and more used in other two areas. One is by using kit screening, it can develop new protocols to expand chemical space. The discovery of new synthetic pathway changed the way scientists thought about designing and building library molecules, which broadened the accessible chemical space and thereby furnished the molecule possessing the biological activity required in future drug discovery. With such higher quality novel molecules, the chemist can increase the success rate and reduce cycle time of heat or lead finding and lead optimization. Secondly, by using high throughput experimental HTE enables the collection of vast reaction data possible for machine learning. More and more companies are using HTE reaction data instead of literature reaction data because of the bias towards only successful results reported in literature. The HTE reaction data is more systematic and covers failed reaction. Thus, it can be better used to train models to predict the performance of a reaction, which will eventually assist the medicinal chemist study. Furthermore, the study to explore dark space of chemical reaction can expand human knowledge of chemistry, which might change chemistry research and drug discovery significantly in the future. In the following, I will introduce one by one. The most important thing for the pharmaceutical industry is to deliver drug candidates in a more cost-effective and timely manner. Library synthesis has been widely and effectively applied to accelerate drug discovery in the stage of target to heat, heat to lead, and lead optimization. During the last 2 decades, WuXi AppTec has developed many enable chemistries and high-throughput technologies and built up a highly productive library synthesis platform to champion the design, synthesis, and testing to accelerate drug discovery programs. WuXi Library Platform is part of WuXi Chemistry Platform, which can support projects from discovery to full development stage for both pharmas and biotechs. On average, an approved drug currently costs $1 to $2 billion and it takes more than 10 years to develop. This is due to expensive and time-consuming wet laboratory experiments, poor initial heat compounds, and high attrition rates in the preclinical phases. Structure-based virtual screening has the potential to mitigate these problems. The WuXi AppTec Virtual Library provides over 15 billion virtual compounds for global research institutes, pharmas, and biotechs. The virtual library is based on rational selected building blocks and reliable chemical designation. The Galaxi tool is the perfect combination of WuXi AppTec's outstanding chemistry expertise and Bisolvite's state-of-the-art software, which allows searching of compounds in minutes, and the synthesis could be completed within 4 to 6 weeks. WuXi AppTec Virtual Library covers a diverse range of chemical space. Diversity analysis was conducted using the classic Merkle framework model. Randomly selected 1 million virtual molecules from the virtual library. These molecules covered over more than 660,000 frameworks with an average of only 1.5 compounds per framework. The total number of high-frequency molecular frameworks was 78, covers less than 3% of the compounds, indicating that the molecules were not concentrated in a limited number of frameworks. It demonstrates the WuXi AppTec Virtual Library has great diversity. Here is an example how to use WuXi Virtual Library to accelerate hit finding and drug discovery. Our client has platform with industry-leading IT tools for machine learning technology, which has wholly owned pipelines and co-developed pipelines with pharmaceuticals as well as biotech companies. The client used their engine to select the virtual compounds from WuXi Virtual Library database and send a request for synthesis. In 2022, more than 10 orders were placed. 3,000 compounds were synthesized and delivered by us. The first batch of molecules for testing were quickly delivered within 2 weeks. The final compounds are divided into 3 copies, 1 copy to the testing platform of WuXi AppTec or third party, 1 copy to the client or the end user, and 1 was retained at WuXi as a backup. With the support of WuXi AppTec Virtual Library Platform, this company signed a strategic collaboration agreement with a global pharmaceutical company. Here is how to get access to the WuXi Virtual Library. Client can contact the Galaxi mailbox to request access to the Galaxi searching tool to do similarity search by themselves, or the compound information could be provided in SMILES file. Client can use CSV file to do virtual screening by their own engine. WuXi AppTec CDD group also has a variety of state-of-the-art software and can help client to screen compounds virtually. The first batch of molecules could be synthesized for testing within 2 weeks. This may greatly shorten the design synthesis testing cycle. WuXi AppTec's compound management team can provide the compound storage and distribution service. As computational efficiency enables screening of trillions of compounds in silico, increasing the likelihood of success, we will continuously expand our virtual space to enhance the virtual screening for pharmaceutical industry. Then for compound collection, library enrichment is still used to support target-to-hit and hit-to-lead stage, but it's also evolved a lot. Not like early days that thousands of compounds per library were made, designers aim for new chemical space and more novel and diverse libraries. Only 200 to 400 compounds designed for each library, and even with multiple different cores. For the designed scaffolds, almost requires no hit found in the literature or patent. That means the chemistry route scoring becomes very challenging, and the majority of the libraries need 8 to 10 step synthesis. WuXi also becomes very important partners to generate library ideas based on more than 20 years experience on drug-like template design and synthesis. About 46% of the library proposal will come from WuXi, which could be run either on the exclusive model or non-exclusive model. This also requires very comprehensive synthesis capability, which could be up to multiple kilogram scale synthesis for scaffold and parallel synthesis of 20-gram scale for K intermediates, and also milligram scale high throughput library production. In recent years, we prepared more than 1,000 scaffolds in 800 gram each, more than 10,000 key intermediates in 10 gram each, and 250,000 compounds in 30 mg each. Quality control results shows the compounds are in very high quality. Compared with the large screening libraries emerged in 1990s, small focused libraries have been the mainstream of focus since 2010s. Today, many innovative capabilities have been developed and applied to library synthesis to make better molecules faster. Parallel medicinal chemistry, PMC, can handle flexible library size from 20 to 200 compounds, deliver products in high speed such as 1 to 4 weeks, and with up to 100% success rate. With the novel protocols developed for parallel synthesis, PMC can now cover a wider scope of diverse reaction types, including standard transformations, ring formation, PhotoRedox chemistry, etc. The main purpose of PMC is to shorten turnaround time of design synthesis test cycle and provide more diverse and novel compounds with limited scaffolds. The de novo design is usually based on HTS results and literature search. After the designed compounds being made, in vitro or in vivo testing will give biological data which will guide the next round of library design. Nowadays, PMC is delivering more and more important impact and enable medicinal chemists to do lead optimization in a fast manner. Here is an example how PMC libraries can accelerate the cycle of design synthesis test. With accumulated parallel synthesis knowledge, this 5-step library could be handled in a parallel format even from the first step. Each cycle focused on optimize one site of molecule, could be executed within 2 to 3 weeks and reach to 75 to 100% success rate. Thus, within 5 months, 6 cycles have been carried out and lead compounds have been found. Compared to traditional Medicam analog synthesis, the PMC library is more efficient with delivery of 17 compounds per chemist per week, and the cost is much lower. Parallel medicinal chemistry was started around 2006. During past 17 years, by endless pursuit of excellence, continuous optimization of workflow, and applying new technologies such as kit screening, PhotoRedox chemistry, microscale synthesis, and high-throughput purification, cycle time has been shortened to 2 to 3 weeks with increased synthetic success rate. Now reached to average 85% success rate, which means with same amount of material, we could provide more diverse compounds much faster to medicinal chemists for testing. Despite the continuous advances in synthetic organic chemistry, medicinal chemists have consistently utilized a narrow range of reactions in the discovery of small molecular drugs. The development of robust protocols where parallel synthesis from widely available building blocks will provide access to novel drug-like motifs. Here is a perfect example to show how to use PMC to develop new protocol to expand the library accessible chemical space, which allows higher quality molecules to be directly accessed via parallel synthesis, reducing the number of design cycles from early hit to clinical candidates and accelerating drug discovery. The two primary goals for this research work are, firstly, enabling the largest and most abundant diversity sources, and secondly, minimizing parallel step count. 4 Medicam approaches reported in the literature show the several disadvantages such as limited diversity source or higher step counts, which limits their application in parallel synthesis. 2 one-port library protocols were developed by PMC to easily access the drug-like branched heterobenzylic amines from structurally diverse aliphatic carboxylic acids via decarboxylative radical amine addition, and from heteroaryl bromides via a parallel lithium halogen exchange amine addition sequence. These 2 protocols offer high analog throughput from widely available building blocks, expanding design space for library synthesis. This article is co-published last year by client and our library team. The second case is the protocol development for parallel Monomer reaction. Heteroaryl halide has very limited availability compared to amines. So starting from amines, where parallel Monomer reaction can increase the diversity significantly. The condition of successful developed library protocol for parallel Monomer reaction is very mild and easy to handle in parallel format. This allows medicinal chemists to design more diverse compounds from available building blocks and quickly make them. It is well known that protocol development is quite time-consuming and need a lot of efforts. It will prolong the overall cycle time for library synthesis if protocol could not be developed in fast manner. Kit screening is a very powerful tool to solve this problem. Our kit screening platform can screen conditions for more than 39 reaction types with 70% success rate. This is based on the failed reactions from bench chemists. The average cycle time of kit screening is 41 hours. In 2022, we have successfully solved more than 3,500 chemistry problems and developed protocols. Condition screening also could be run in microscale with less than 1 mg of sample for each condition. It can save precious material for medicinal chemists to develop protocols. Here is an example for unsuccessful backward reaction. 24 conditions with 6 different catalysts and 2 solvents and 2 bases were tried at 120 degrees as first round screening. The product was formed in 70% by 1 condition, but epimerization happened. Then second round screening was carried out with 12 catalysts in 2 different solvents on the lower temperature, 40 degrees. By T-butyl X-Force Palladium G3 catalyst, it gave product in 70% yield without epimerization. This protocol was developed within 3 days and used for library production successfully. Kit screening is a very powerful tool to solve the problem quickly, improve the success rate, reduce the cycle time, as well as develop a new protocol for both library and Medicam synthesis. The other technology is high-throughput experimentation, HTE. Nowadays, more and more machine learning companies need vast of data to expand the chemical space or explore the dark space of chemical reactions. HTE is used to generate the reaction data by executing a large number of experiments in a short time frame, instead of only literature reaction data. The literature reaction data is considered to be pervasive and long-lasting bias towards only successful results. When such data is fed into machine learning, it may generate a biased algorithm that will lead to poor reaction prediction performance. On the contrary, the reaction data from HTE experiments is more systematic, reliable, and covers both successful and failed reaction. Thus, it can better used to train models to generate algorithms that has significantly improved the predictive performance, which will eventually benefit expanding chemical space. WuXi AppTec's library team applied the HTE technology to explore the dark space of chemical reactions. We can finish more than 10,000 HTE reactions within 4 weeks, including reaction matrix design, reaction setup, data collection, and analysis. We have state-of-the-art software to process and analyze LC mass spectrum, which can handle 384 LC mass spectrum in 10 minutes. Here shows the workflow. Generally, whole process of 384 reactions could be finished within 72 hours, including reaction matrix design, reaction setup, workup, data collection, and analysis. With the analyzed data, next round of reaction matrix will be designed and executed. By vast of reaction data generated and the evolution of computational capability, it is predicted the next 50 years there will be breakthrough for molecular editing and machine learning. PMC is also used to accelerate the research of some hot areas. Covalent drugs have experienced a significant renewed interest in drug discovery. This resurgence has been accompanied by a better understanding of the reactivity relationships required to engage selective covalent bonds between nucleophilic proteins and electrophilic small molecules. Amgen discovered and developed a covalent inhibitor of KRAS G12C, sotorasib. A lot of efforts have been made by either biotechs or farmers to find highly potent and selective covalent inhibitors. Library synthesis has been a very useful tool to support medicinal chemistry research employing covalent chemistries. Generally, covalent warheads serve as electrophiles which react with nucleophiles, such as a synthesizer in the biological system to form covalent bond, resulting in superior activity. Cysteine-targeted covalent chemistry has been indisputably fruitful for delivering safe and relatively selective drugs. Nowadays, a lot of novel warheads were rapidly emerging within the scientific literature, aiming for expanding the toolkits of reactive modalities that are amenable to chemical biology and drug discovery. WuXi AppTec has plenty of experience on developing new warheads for covalent libraries with more than 50,000 compounds delivered in recent years. Here are several cases that using library tool, it can convert hundreds of nucleophile molecules to covalent compound within 2 to 4 weeks by high success rate. The third case showed the bicyclobutane carboxylic amide as a new class of reactive electrophiles for selective and irreversible inhibition of targeted protein. By tuning BCB amide reactivity and optimizing its disposition on the ligand, the author obtained a selective covalent inhibitor of BTK. We continue following the literature closely and validate and recommend new warheads to medicinal chemists. Another hot area, PROTAC. The advantages of PROTAC are well known as it can degrade the target proteins specifically with selective binders. It targets undruggable proteins and requires low dose or concentration and prolong the duration of action. However, chemistry difficulties, compound stability, purification challenge, and long synthetic cycle time also are the challenges for PROTAC research. WuXi AppTec has plenty of experience on the synthesis of PROTAC libraries with more than 30,000 compounds delivered in recent years. Here we use several cases to demonstrate how PMC could accelerate PROTAC research. The first case is the modification for E3 ligands. Different synthetic strategies such as parallel microwave reactions and PhotoRedox chemistry were used to produce 228 different E3 ligands within 2 to 3 weeks with 76% success rate. Different E3 ligands could be done derivatization in parallel format, thus increase the speed of PROTAC research a lot. The second case is using 57 different linkers and 2 E3 ligand to generate 97 PROTAC molecules within 6 weeks by 228 steps. The final compounds were prepared with 3 steps: amide formation, debulk, and SNAR displacement with more than 1,000 molecular weight. We have dedicated a purification team to support PROTAC library purification. They developed more than 200 conditions for the challenge PROTAC compounds purification and QC. In this case, a very good success rate of 85% was achieved. For the third case, the challenge is to make novel peptide linkers for PROTACs. To speed up the linker synthesis with 8 steps and make purification easier, we developed a protocol of solid phase synthesis for this library, which only require washing of the beads and achieved a 100% success rate within 6 weeks cycle time. We are still investigating and applying parallel synthesis strategy into other hot areas for drug discovery and try to further speed up the lead optimization stage in more cost-effective way. Hopefully, we can share more progress in near future. Then, in the second part, I'd like to introduce what are the critical factors for PMC. This also demonstrates how we optimize PMC process to speed up the medicam research. The whole process includes 4 stages. The first stage, reagent acquisition, also takes 1 week, which is already 25 to 30% of the total cycle time. Reagent acquisition speed is very critical to the speed of PMC library. In WuXi, the warehouse stored 50,000 monomers, which could be obtained within the same day or the second day. This storage list was often asked by medicinal chemists to use it to design their PMC library. Furthermore, WuXi Lab Network also can sourcing for 220,000 reagents within 3 to 4 days, which ensure the quick start of PMC library. We also provide management of the monomers synthesized or purchased for clients exclusively to further reduce the monomer acquisition cycle time. For example, for this project, we managed about 6,000 building blocks for clients, and about 4,200 building blocks. 70% of the total were reused for library synthesis for past 5 years. By this model, it not only reduced cycle time by 33% as the majority of the building blocks are in storage, but also reduced the cost of whole library by 35%. The second stage for PMC is synthesis, which includes chemistry validation and library production. More than 46 reaction types are widely used for PMC to generate diverse compounds. Protocols well developed to run some reaction types which seldom applied in parallel synthesis before. For example, by using solid-supported reagents such as polymer-supported triphenylphosphine, it can enable Wittig reaction in parallel formats. By palladium catalyst and ligand, the carbonylation also could be used for library synthesis. By best-man or hero reagent, it can in situ generate alky from aldehyde parallelly, then used for click chemistry, which expanded the compound diversity significantly. More cyclization reactions showed here to form drug-like cores in parallel format for Medicam projects. Recently, PhotoRedox chemistry also used significantly in PMC library to reduce synthetic steps and do cross-coupling more efficiently and innovatively. In 2013, Professor David Macmillan published the article, Visible Light PhotoRedox Catalysis with Transition Metal Complexes, Application in Organic Synthesis. It brought a lot of new ideas for organic synthesis route scouting and also brought many possibilities and faster approaches for PMC library design. We have established from microgram scale for PhotoRedox condition screening capability to milligram scale PMC PhotoRedox library capability to gram scale scaffold synthesis capability. Combined with flow chemistry, it also could be scaled up to even kilogram scale. Here are two examples to apply PhotoRedox in PMC library synthesis. The left one is decarboxylative coupling reaction, which now widely used in the synthesis for Medicam projects. The condition of this reaction is mild and can cover wide substrate scope with high efficiency. And medicinal chemists can use this for the later stage functionalization on their drug-like cores. We have different PhotoRedox parallel reactors to run 100 reactions very easily. The case on the right is the carbon-carbon cross electrophile coupling, by which the halide reagents are easily to get, and this reaction gave high success rate and reproducibility. While by traditional organic synthesis, it might need multiple steps. This gave medicinal chemists freedom to do derivatization at final step. This case showed the PhotoRedox combined with flow chemistry to produce scaffold for library synthesis. In the literature, the batch reaction reported with very dilute reaction solution. It is very difficult to do scale-up by literature procedure. We assembled in-house flow system on the blue LED light. The scaffold could be scaled up to 50 grams, which is enough for library synthesis. By this way, medicinal chemists can get more novel scaffolds very quickly for their research project. Automation of synthesis operation is attracting more and more attention in recent years, allowing increased throughput while decreasing the cycle time. To improve efficiency, we co-invented an automated parallel synthesis equipment with a local leading robotic company. Compared with manually, it has higher reproducibility as the synthesis chamber is nitrogen protected and the dispensing deviation is less than 5%. Furthermore, it is 24 hours and 7 days on standby to increase the efficiency of library production. As an example shown below, it can reduce the half of the synthetic cycle time while providing higher success rate. As the purification and the QC process were well established in a high throughput way, we will not talk about it today. For the final stage of PMC is compound management. It includes generating solid copy or solution copy for different purposes, compound storage, and tracking. The slides show we can collect and store compounds from different partners or collaborators and manage them at WuXi. By the instruction from clients, we can generate assay-ready plates and ship to assay providers. This is to enable any assay anywhere. The Titian mosaic system or WuXi's compound tracking system could make the compound amount or compound status visible to client exclusively. If the chemistry partner and the assay partner are all in China, the cycle time from compound ready to start testing is only 1 or 2 days, which significantly reduces the logistic cycle time, at least by 5 to 7 days. We can handle very complicated workflow for compound management. In this case, we make 9 copies of acid-ready plates and 2 copies of solid. The solution copies were delivered as dry ice shipment to 2 sites, used for assay immediately once clients receive them. The solid copy could be used for storage. We passed the client audit without any issue found. As a summary, to effectively accelerate drug discovery in the stage of target-to-hit, hit-to-lead, and lead optimization, we have built a virtual library, library enrichment, and PMC capability to establish WuXi library platform. Recently, library technology is not only used to generate compounds, but also to develop new protocols, to expand a new chemical space, and to generate vast data to explore dark space of chemical reactions. New technologies such as HTE, automation, and new analytical methods are accelerating the discovery of new reaction methods. Further, integration of computational reaction modeling with the vast quantity of experimental data generated by nanoscale HTE has the potential to build more informative models that can predict the successful reaction conditions or even discover new reactions. Advancement in the synthetic chemistry are certainly to remain highly relevant to the mission of inventing new medicines to improve the lives of patients worldwide. Parallel chemistry is playing more and more important role to further support drug discovery in more timely and cost-effective manner. At last, I would like to thank my colleagues, Dr. Yu Gao and Mr. Li Qiangqu, who also have more than 20 years library experience and contributed a lot for this presentation. Also appreciate my colleague John for his introduction and full support for our work. Thanks a lot for your attention.
Thank you, Yan, for an interesting presentation. We now have time for some questions. Question 1. I'm pretty interested in using HTE for reaction prediction. I believe it can reshape the way of medicinal chemistry and drug discovery. Do you have any more information or experience on that?
Thank you for the question. Yes, we have experience of working with some clients for HTE data generation and processing since last year. The first step is data generation. HTE involves running a large number of experiments in parallel using automated workflow, allowing rapid screening of various reaction conditions, catalysts, and reagents. This generates vast amount of data on various chemical reactions, many of which might be previously unexplored. The second step is data processing. Compared with literature data, the data generated from HTE is more systematic and covers field reactions. Thus, it can amend the pervasive and long-lasting bias towards only successful results reported in the literature. Data processing involves removing noise, handling missing values, and converting data into structured format suitable for AI analysis. And the third step is modeling training, which mostly done by the clients. This is to train models such as machine learning or deep learning algorithms on the processed data to learn patterns and the relationships between the parameters and reaction outcomes. The models can help predict the likelihood of success, yield, or selectivity of unexplored reactions. Also needed is to validate the models on a separate data set to ensure their accuracy and generalizability. Refine the models as needed based on their performance. Then the last step is reaction prediction and further improvement. Use the trained models to predict the outcomes for new unexplored reactions. This can guide further experimentation and help identify novel and promising reactions in the dark space. Continuously update the models with new experiment data to improve the accuracy and the predictive capabilities. This iterative process helps explore the dark space more efficiently and effectively over time.
Thank you. Let's see, question 2. Thank you very much for this outstanding webinar. Can you share any specific novel techniques or methodologies in library synthesis that have significantly accelerated the drug discovery process?
Yeah, parallel medicinal chemistry is a very powerful technique in library synthesis that can significantly accelerate drug discovery. The main purpose of PMC is to shorten turnaround time of design, make, test cycle, and provide more compounds from limited scaffolds for biology tests. WuXi AppTec has more than 20 years experience on library synthesis and developed a streamlined workflow, accumulated parallel synthesis knowledge, as well as flexible adoption of new technologies such as catalyst screening technology, PhotoRedox, microscale synthesis, and purification technology. We have effectively applied the PMC model to champion the design, synthesis, and purification of target molecules to accelerate drug discovery programs. Typically, for one project, if by PMC model, 50 to 100 diversified compounds can be quickly synthesized within 2 to 3 weeks, which can significantly speed up the heat and lead finding and lead optimization. Parallel medicinal chemistry has been an essential part of drug discovery process for both big pharmas and quite a number of biotechs.
Great, thank you. Question 3 now. I'm very impressed with your capability of developing new protocols for library synthesis. Do you use the same reaction condition for all monomers? Can you please comment on how you validate the protocol for different building blocks? I ask this because we usually find one protocol is not suitable for all design building blocks.
Yes, this is a good question. We also find in a lot of cases that one protocol could not cover all building blocks designed in one library. This is because the diversity of the building blocks. The development of new protocol for PMC library will provide access to novel drug-like molecular and expand the chemical space. Thus, it is of vital importance and can help accelerate drug discovery. At WuXi, we have developed a very streamlined workflow to do building block classification based on the structures, reactivities, as well as our rich experience. Then we use the technologies such as kit screening, HTE, PhotoRedox, etc. to validate different conditions for different building blocks to find suitable protocols for each cluster. This can improve the success rate and increase the compound diversity significantly.
Thank you, Yan. Question 4. I'm very interested in your PROTAC library and Covalent library capability. Can you comment on your availability and experience?
Sure. Indeed, the PROTAC is a hot topic in recent years and draws a lot of attention. WuXi has started to build PROTAC capability since 2015, so we already have more than 7 years experience in this area. We are capable of carrying out 1 to 5 step parallel synthesis and deliver final targets in 2 to 4 weeks fast turnaround time. We also have in stock more than 150 reference-reported E3 ligase linkers, including PEG-alky click reaction chain rigid linkers, as well as precursors of linker plus E3 ligand. This readily available study material can ensure quick start and fast employment of the research work. Purification of PROTAC is also very challenging in many cases due to the limited solubility, poor stability, and the high polarity of this molecule. WuXi has dedicated the analytical purification team for PROTAC compounds. The team has developed more than 200 different purification conditions, include columns, modifiers, and gradients, and delivered more than 80,000 PROTAC library compounds. For Covalent library, which actually has more than 7 years experience in this area, we have developed the library production protocols for more than 30 warheads, as well as high throughput purification capability for Covalent library. We have delivered more than 50,000 library compounds. We're capable of carrying out 1 to 5 step parallel synthesis and deliver final targets in 2 to 4 weeks fast turnaround time. If you would like to know more about our capability, please feel free to contact us. We can share more information offline. Thank you.
Thank you. I'm afraid that's all the time we have. If we didn't answer your questions, the speakers will follow up directly. Thank you again, Dr. Zhu, for the insightful presentation, and thank you, participants, for being a great audience. I will turn the call back over to CNE.
I'm afraid that's all the time we have, and thank you again, Dr. Yan and John, for a fascinating presentation. And thank you, participants, for being a great audience. Please be sure to check CNN or CNN Online for the replay of this webinar and information on the next edition of CNN webinars. Our thanks to On24 for technology and production services, and thank you, WuXi AppTec, for the sponsorship that made this interactive webcast possible. For CNN webinars, I'm Ann Thayer. Hope you have a good day.